welcome you all to the Week 7 live interaction session for the course Environmental Biotechnology for the course ID NOC 25 BT 83. It's already 5:05, so we will start the session now. Is it my slide and PPT visible?
Yes, sir, but your voice is breaking, sir.
Okay. Now it is fine. Hello. Now it is fine.
No, sir, still it is breaking.
Is it fine?
Yes, sir.
Okay. Welcome you all to the course Environmental Biotechnology. I hope you have all finished your assignments. So in today's class, we will discuss on week 7 live interaction session. So here we will be dividing the session into two. The first, we will see the recap of the important concepts that have been covered in week 6 and week 7. For the MCQs, the sample MCQs for the week 6 and 7. So myself, Ajit Kumar V, a PhD research scholar from the Bioscience and Bioengineering Department from IIT Guwahati. Today I will be giving a recap of week 7 session for the course Environmental Biotechnology for the course ID NOC 25 BT 83. So moving on to my personal introduction, myself Ajit Kumar, doing a joint degree research program, a collaboration research between IIT Guwahati and IIT BHU Varanasi. I work in a laboratory of biochemical and environmental engineering laboratory where our lab focuses on wastewater treatment approaches via different techniques. And my research inclines on applying the enzymes, particularly in the wastewater treatment. And moving on to my qualification, I've done BTech Biotechnology from Sastra University and MTech Industrial Biotechnology from NIT Surathkal, Karnataka. And moving on to my work experience, I worked in many pharma companies like Biocon and Enzym Bioscience. So this is all about my personal introduction. So without any delay, we will move on to uh, the recap of the week 6 and weeks lectures of the environmental biotechnology course. So in the last class, the last live interaction session, we have covered some important concepts such as what are all the different carbon and energy sources that are available for the microorganism in an microbial ecosystem. And also, we have learned the role of selective pressure and how it drives the microbial ecosystem, how it helps the microorganism to evolve in the evolutionary. And also, we have seen different growth strategy like R and K selected growth strategy. And the most important concepts like thermodynamics, where especially oxidation and reduction reactions are most important for carrying out their metabolism. And we have seen how the logic of electron concepts evolve and how the electrons has been transferred from the substrate to the microorganism, which helps in driving their metabolism. And moreover, we have seen the important concept of catabolism and anabolism. We have seen the technical terms and logic behind it, and we have seen various metabolic pathway that involves the catabolism and anabolism in a microorganism. And we have ended the session with the syntrophy, the importance of syntrophy, and various examples in the environment. So these are all the topics that we have covered so far. in the last session. So today we will be seeing some more important concepts, mainly the methods that are involved in measuring the microbial ecosystem. So in today's session, moving on to the methods, first we will introduce the term and fresh up our mind with the microbial ecosystem. So what is microbial ecosystem? Ecosystem is nothing but it is a sum of both biotic and abiotic factors. And we all know that biotic factors means all the organism. It can be plants, animals, microorganism too. In microorganism, it can be either prokaryote, eukaryotes. So all the biotic factors and the abiotic factors. Abiotic factors includes it can be temperature, pH, soil content, moisture content, anything which is non-living matter. So measuring the both or summing up both biotic and abiotic factor in a particular environment, we call it as an ecosystem. So it is an not a static process, it is dynamic. That means it changes as the time evolves in the evolutionary. So it is a dynamic complex that occurs between the biotic factors, which can be plant, animal, and microbes, and their interaction with the abiotic surrounding. So ecosystem will contain a different habitat. Habitat is nothing but a place that we live. Like we used to live in a home or house, right? So like that, for an ecosystem, we call it an habitat where it refers to the zone in which a particular species or population they naturally live and grow. So we call it as the place or the home for the ecosystem. So here you want to have an importance or know the terms like ecosystem and habitat, which is very important for further studies. So how do we measure this microbial ecology? What are all the major components that we study in the microbial ecology? The first one is their biodiversity. Biodiversity means it contains all the living organism, right? So biodiversity, how do we estimate it? First, you have to identify them and we have to quantify them. What are all the microorganisms that are present in a particular habitat? So how do we identify them? So how do we identify them? By using the 16S rRNA approach through various techniques. And quantify refers to how much microorganism, particular microorganism are present in their habitat. So if you take any environment, it can be a soil or water environment. So if you consider soil environment or a soil ecosystem, you could observe a millions of microbes that are present. So first, your role is to identify what are all the different microorganisms that are present in a particular environment and also quantify or try to find the abundance of microorganism because, for example, in a hot temperate region, you could find a thermophilic microorganism dominates over the other type of microorganism. This microorganism can be a saccharophilic microorganism or mesophilic microorganism. So, depending upon the region, this microorganism abundance will vary. So, this is why it is very important to identify them as well as to quantify them in order to study their biodiversity. And the second most important component is their microbial activity. After identifying and finding their abundance, we have to study the individual microorganisms' metabolic activity. And we have to measure the metabolic activity that are carried out by them in the habitat. So why it is so important? Because if you are measuring their enzyme activity, for example, one of the metabolic activity is there measuring their enzyme activity and what are all the products that are secreted by the specific microorganism. By knowing their metabolic activity, we could use it for our environmental application. So these are all the two major components that you have to see when you are studying the microbial ecology. First is their biodiversity and second is their microbial activity. And if you have gone through the early lectures of the environmental biotechnology course, You could have come across these four questions. And at this point of time, I know you know all these questions and how to answer it. So we will brush up it with a recap. So first question that you have to ask is who is here? That is, what are all the microorganisms that are present in a given ecosystem? And the second question is what can they do? What can they do? That is, what role they are playing in a given micron in a given ecosystem and what they are doing. So actually, their activity can be many. They can secrete 10,000 types of enzyme activity, but for a particular environment and for a particular environment, based on their role that they have been given in a particular environment, what they are doing now. For example, if suppose a microorganism is isolated from the textile effluent, their current role will be what? They evolve or adapt to the environment where a similar group, a particular group of enzymes will be secreting. So, based on their environmental condition, these microorganisms will play differently. And the last question is how do they interact and work as a system? That is, how these microorganisms interact among themselves. Either they are cooperating each other or they are showing a parasitist mode or mutualistic mode or antagonistic mode. So depending upon their mode of interaction among themselves is also very important and also with their environmental activity also. What does it mean? So not only they interact with each other but also with the environment. So you have to understand their interaction part as well. So if you can confidently answer these questions for a particular environment, then your understanding of microbial ecology is strengthened. So these are the four basic or fundamental questions that one needs to ask to understand the surroundings or environment of microbial organism. So in order to refine it, what environmental biotechnology sees. The first, if you see an environment, first question is whether what are all the type of bacteria or archaea. It doesn't need to be like strict to the eukaryotes. It can also extend to the fungi and protozoa as well. First question is what are all the microorganisms? It can be bacteria, fungi, protozoa, yeast, algae. They are present in the given sample. Next, how many different types of that is taxonomic as well as the physiological. What does it mean? What are the different species of microorganisms that are present and what are their physiological role that they play? This is also and very important because as particular type, for example, uh, if you want to treat an heavy metal contaminated site, that means you have to, for example, the heavy metal can be arsenic. So if you take the arsenic example, you will be having a specific group of microorganisms that can assimilate this arsenic and convert into non-toxic form. So finding their type taxonomically and physiologically also helps us to understand the environment better and which microorganisms are active and growing. So among the different microorganisms, we have to identify which is actively growing for participating in the given environment. And what's the ecology of microorganism in the context of their environment? How do they interact with the environment? And what are all the ecology of that microorganism is also very important while studying the environment. And most importantly, after understanding the first four question, you have to ask yourself how we can apply this knowledge for environmental application. It can either be a bioremediation application or for fermentation or any industrial product formation or we can use the microorganism for important chemical production also. So these are all the five important questions that one need to ask. First, as I repeat, first is you have to identify the microorganism and you have to find how many different types of species that are existing in the microorganism and among these species which is actively growing and participating. in the environment. And you have to understand the ecology with respect to the environment, and also you have to think how we can use this microorganism for our environmental purpose so that it satisfies the application part. So these are all the most of the fundamental questions that one needs to ask, and the next is, if we move on to the method of the microbial ecology, the first and most basic method is their isolation of microorganism. It can be, we will be seeing the isolation technique in the further slides, but the first and most, the great anomaly is that the great plate count anomaly, which is nothing but, first we will see in a layman perspective. For example, if you in a WhatsApp, I think you all have WhatsApp, right? So in that you have various groups. So in each groups there may be 100 to 1,000 members. But if you put a message to that WhatsApp group, only 10 to 15 people may respond to that message depending upon their busy schedule, right? So likewise, likewise, if you see the microorganism after separating from the soil, if you observe them in the microscope, you would see 100 to 1,000 colonies. I will explain the process. For example, if you take a 1 gram of soil and if you dilute it in an 100 ml, which is an 10 to the power of minus 2 dilution, and you take 10 ml of this first diluent and transfer into the next 90 ml, and it is an 10 to the power of minus 4, and proceeds after, you will, after proceeding to the 10 to the power of minus 5, you will be viewing in two different aspects. One is the microscope, another one is spreading onto the agar plates. So if you spread the culture on the agar plate versus micro and view the microorganism on the microscope, what you will observe is that you will observe 100 to 1,000 cells, but on the agar plate you can see only 1 percentage of the, or even the less than of what you observed in the microscopy. What does it implies? It implies that only few microorganisms can grow. And what happened to the rest? Because since we have not given an appropriate environment and nutrient condition, they are not able to grow it. Whereas here, as we are using the global staining technique, it, it used to stain all the cells. But these all the cells cannot be cultured, right? That is why what happens here is, if you are not providing appropriate condition or environment to the microorganism, you won't be able to see. That is why all the microorganisms cannot be cultured as easily. That is why we call it as great plate count anomaly. That is, what you see is not always a full picture, right? So in microscope, you could view 100 to 1,000 cells, but if you translate them into the agar plate and once you plate it, you only see 10 to 15 colonies. That's because the nutrient media and the condition that you have provided, that only favors the growth of a particular group of organism, not the all microorganism. That is why we say that all the microbes cannot be cultured. And in the next slide, we will see what are the classification of microorganism based on the culture. So if you see the categories of culturability of the microorganism in the nature, you will see the first is the cultured group of microorganisms. As the name implies, these microorganisms that has been successfully isolated and purified in the laboratory. How? By providing the appropriate nutrition condition and the environmental condition, like giving appropriate temperature, pH, and other factors. So once it has been successfully isolated and purified, we will be depositing in the culture collection centers. What do we give? We give the information about the isolated microorganism. So this serves as the model organism for the rest 99 percentage of what is known about the microbiology or environmental biotechnology. So what do we call the culture? That has the cultured microorganism is nothing but those that have been successfully isolated and purified. So these will be deposited in the culture collection center, and these serves as the model organism for the researchers to study on. And the second category of the microorganism, which is uncultured. What does it mean? The one which have no appropriate growth medium has been devised. That is, till now they have not been identified the correct proportion of nutrient media as well as the environmental condition. So in that you have two classification. One is culturable group of uncultured organism and unculturable group of uncultured organism. So first we will see a culturable. Culturable means it is a if you provide an appropriate growth media and if you could find the correct combination of growth media as well as the environmental condition, these microorganisms can be cultured. Whereas in unculturable, the microorganism whose physiological state prevents them from being grown. So here we could not identify the correct proportion till now. That is, researchers are trying to find an appropriate condition to find a way for unculturable microorganism. So these are all the categories of culturability for microorganism where first cultured, it is already cultured and it has been deposited in the culture collection. Whereas in uncultured, scientists are trying to identify the microorganism which are culturable and non-unculturable. So these are all the categories of the microorganism. And if you see the broad outline, and if you see the broad outline, the methods of the microbial ecology, first, as I said, first identify the microorganism. Here you have to use specific tool. It can be either direct isolation or you can use the enrichment mode of isolation. We will be discussing in the upcoming slides. And after identification, after isolation, you have to identify them. How do you identify? Through 16S rRNA sequencing approach, where you could identify what is the microorganism name. And also you can characterize the biochemical, physiological, and ecological triad. Here you could use both qualitative and quantitative tools and identify what could be the group of microorganisms that they characterize. There are numerous biochemical characterization techniques that are available for a given microorganism. For example, if you are trying to isolate an oxygen tolerant microorganism, then you could go for a thioglycolate based approach. What happens here? It tells us whether your microorganism are aerobic, anaerobic, facultative, or microaerophilic. This is one of the tests for oxygen tolerant if you are aiming for an oxygen tolerant microorganism. And third is molecular or genomic analysis. What happens? Here you could do molecular analysis by PCR based technique or metagenomic strategy. So these are all the broad outlines for studying the microbial ecology. And if you see the selection method, there are two types of selection method. One is cultural cultivation dependent and culture independent technique. This is to study their biodiversity, that is first identifying and knowing the abundance of the microorganism, or you could study them, study their metabolic functions, or sometime both. So this is the selection method, and depending upon the requirement, we will be going for them. And if you move on to the isolation of pure culture of the bacterial strain, what do you mean by pure culture? The pure culture signifies isolating a single strain of microorganism. So here you will not expect any other microorganism or undesired microorganism. So you will have only one type of microorganism or a single strain of microorganism. And how do you isolate this pure strain? First, you will be taking a small amount of sample. It can be a water sample, soil sample, or wastewater sample too. So, for example, if you are taking a soil sample, you can take 1 gram of soil sample, and if you are and the next step is you have to decide whether you are going for a direct isolation or enrichment mode of isolation. If you are going for a direct mode of isolation, what you will do is you will take 1 gram of soil and you will put it in a water or a normal media, and you will incubate the sample for an appropriate amount of time. And once the growth has been achieved, you will be spreading onto the agar plate where you will be giving appropriate nutrient condition and the environmental condition. Once you have spread onto the agar plate, you will see an colony, and next step is you will be isolating a single colony from the plate, and you will be subculturing. Subculturing is nothing but you will be taking the single colony and you will be again spread onto the agar plate. We call it as subculturing. This is the direct mode of isolation of pure culture. But if you want to go for an enrichment culture, the term itself signifies it is enriching a particular type of microorganism. For example, if you are looking for a um, if you are looking for some kind of like arsenic remediation, you will first what you will take an arsenic contaminated wastewater or soil sample. Then you will add an arsenic metal into the media which contains the soil. So what happens? The microorganism which can tolerate the level of arsenic will grow. So we will be spreading onto the plate and we will observe for the colonies. And we will test these colonies for the arsenic tolerance. So what happens here? We will be varying the concentration of the arsenic in order to see how much level of arsenic it can tolerate. So by this we are enriching a particular type of microorganism which can tolerate the arsenic level. And once it has been successfully obtained the single colony, we will be subculturing it for future purpose. So these are the two main technique for isolation of pure culture, where in the first technique you can go for direct isolation, or you can, depending upon the research interest, you can go for enrichment culture mode of technique. And if you see the enrichment culture, the isolation here requires separating different organism from the microbial community. Whereas in the pure isolation, whereas in the enrichment culture technique, what happens here is it prefers a particular growth and selection, that is isolating a specific group of microorganism, and we will be neglecting the other group of microorganism since we are giving a particular nutrient condition. So here the selection of the population where with one dominant strain or one dominant microbial group, which is accumulated in the system of cultivation because of the environment or the selective pressure that we are giving to the community. For enrichment, you need two important criteria. One is an appropriate media components, and next is you have to give an incubation condition that are selective for a desired microorganism and counter the selective for undesired organism. This implies that you will be giving a selective pressure for your interest of microorganism so that the undesired microorganism will not grow in the particular media. And and what are all the selective condition? What are all the selective condition that normally one observe for? First is the source of energy. What kind of energy the microorganism demands, either ATP, NADH, or NADPH. And second is the source of carbon, whether it is utilizing a simple sugar like glucose, or it uses CO2 as a carbon source, or complex sources like dextrose, fructose, or etc. So depending upon the source of carbon, you can select the microorganism that you are focusing on, and source of nitrogen and phosphorus. Some microorganism can some microorganism can survive in the presence of ammonia. If you want to focus on that microorganism, you can provide an high amount of ammonia to the media and you can selectively select the microorganism. And depending upon the temperature, as we have already gone through in the earlier lectures, depending on the temperature, there are different types of microorganism. One is thermophilic microorganism, which can tolerate higher temperature, whereas the second class of microorganism where where sacrophilic type of microorganism where it can tolerate at very low temperature, whereas mesophilic will try to thrive in the normal room temperature. So this is also one of the criteria when a scientist are focusing on particular group of organism. Then is pH. As we know, there are three kind of pH. One is acidic, alkali, and neutral pH. And if the scientists are focusing on the acidic group of microorganism, in that case, you can provide the acidic pH type of nutrients for the selection. And if your focus is on alkali, you can provide a pH range around above 6, above 7 to 14. And if you are focusing on neutral region, you can adjust the pH to 7 and you can start your selection process. Then, depending upon the concentration of the heavy metal, as I said previously, if you are focused on some heavy metals like arsenic, you can provide the arsenic to the media and you can test which microorganism are growing and how much it is tolerating in the given environment. And the next is you can provide a specific antibiotic intermediate to selectively select the antibiotic resistant microorganism. And you can also use the concentration of dissolved oxygen. As I said before, if you're focusing on oxygen tolerant microorganism, you can go for like aerobic mode of microorganism, anaerobic, facultative, and microaerophilic microorganism, depending upon the oxygen that is supplied to the nutrient media. And if you want to focus on osmotic pressure, microorganism should handle higher osmotic pressure. You can provide osmotic pressure, and depending upon the spectrum and intensity of light, you can also segregate and enrich the culture. So these are all the some of the conditions that you can provide. Not only individually, you can use the different combination of this selection criteria in order to select a microorganism for your experiments or for your research purpose. So after identifying the microorganism, the next objective is to quantify the microbial biomass. And how do you quantify? Quantification is nothing but you have to identify and determine how much number of cells that are present in the biomass, or you have to quantify the cell biomass. There are different strategies or equipments that are available. One is you can view in the microscopic, which is common one, or you can use the advanced technique like flow cytometry in order to evaluate the number of cells. And also you can measure the physical measurement can be done for the cells as well as you can go for physically measure the cells as well as the biomass concentration, or you can chemically measure through various assays for measuring the cells and biomass concentration. And we'll be seeing some of the methods how it is measured. And third and fourth is you can use biological method for enumeration of cells and virus. Enumeration is nothing but identifying the cell number. And the fifth is you can measure the biomass physiologically and molecular biological techniques also. So these are all the some of the approaches that you can follow for quantification of microbial biomass. And if you see the technique that are followed for measuring the biomass is one of the technique is most probable number technique. And what happens here? Here you will take water or wastewater or a solid sample. Here you will be giving an enrichment media and you will be adding the gram of solid or liquid. You will allow it to grow for some time and you could see the growth in the diluted sample. What happens here? Here you will add the 9 ml in each flask, in each test tube, and you will be adding the first 1 ml of direct this enrichment culture media into the first flask. Here, what happens here is you will observe a high density level of growth, and you just take 1 ml of this and transfer it consequently. And at one point, you will see that there will be no growth. How and why? Because the microorganisms has been diluted so that at one point of dilution you will not observe any growth. So what happens here? You will be serially diluting the inoculum until the final tube shows the no growth. Why it is? done because in this case you can take 10 to the power of minus 4 tube so that you could observe a very less number of microorganisms. And once you plate this 10 to the power of minus 4 tube onto the agar plate, you will observe very less amount of colonies, which helps us to isolate the pure culture. So normally it is used for isolating a specific group of microorganisms from the diverse environment. And the most probable number can be done using the highly selective media with the right proportion of incubation condition to target a small group of microorganisms. So this is one of the important technique for evaluating the pure culture. And what are all the alternative techniques that are used for isolating the pure microbial culture? So first is you can separate them mechanically using the micro manipulator. What happens here? They they used to physically separate the cells using the fine needle or tweezer. So this is like mechanical or physical mode of separating the cells. And the second method is flow cytometry. What happens here? They will try to sort the cells based on their size or fluorescence or the signal that they generate from the cells. So sorting the cells from the group of cells can be done using the flow cytometry. And the third technique is the magnetic or immunomagnetic separation. What happens here? The microbes will be magnetically tagged using some antibodies, so their separation will be more selective. This is a more advanced level of separating the pure culture. And fourth is cells chromatography. What happens here? The microbes will be passing through a column where the separation will be taking place based on some criteria or property that they have. And the last one is laser quizzes. What happens here? The laser beam will allow us to trap and move the single microorganism without even touching them. So these are all the some of the alternative methods that can be focused on for isolation of pure microbial culture apart from the most probable number technique. And the next is the culture purity. How do we ensure the purity of the culture? First, in an axenic culture, first you have to understand the term axenic culture, which is nothing but we call it as a pure culture, a culture that is free from the undesired microorganism or the contaminant. If the culture doesn't contain any contaminants, we call it as the condition as axenic culture. So, how do we ensure the purity of the isolated culture? It is through the combination of three aspects. One is the microscope. What do we see through the microscope? We look for the same size. If you take a culture and if you want to say whether it is pure or not, you will stain the culture and observe through microscopy. And if you see all the cells are of same size or same shape or having a same staining pattern, then you can say that the microorganism is highly pure. And second is you can observe the characteristic of the colony. Here you can look for whether the all colonies are identical. For example, if you are working on the bacillus, you will observe the rod-shaped bacteria if under the microscope, and if you are observing some spherical shape or chain-like structure, then your culture is contaminated with undesired microorganism. And third is testing the culture for their growth in other media too. So what happens here? You can test the same culture in the other media, and if you are able to observe same behavior, then you could say that the purity your culture is highly pure. And what are all the qualifying criteria that we keep? First, it should have uniform staining characteristics. Second point, it should have the uniform colony and self morphology. And third is, it should not contain any contaminants or undesired microorganism or foreign microbes in the growth test. So this is how you measure the culture purity, and these are all the some of the qualifying criteria that you need to keep on the mind while working on culture purity. And as I said, um, in order to go for an isolation technique, there are two main categories. One is culture dependent approach, next is culture independent approach. In culture dependent approach, as the term suggests, you need a culture in order to isolate the microorganism. Here, what happens? First, you will take an environmental sample. It can be either soil sample or a water sample. You can go for either direct isolation, enrichment based isolation. As we have seen the criteria, how direct isolation is taking place and how enrichment is taking place. Once through either of the technique, you have identified the isolated the microorganism, and through previously you have identified that pure it is a pure culture, and you have identified the pure culture by going through the qualifying criteria that we have mentioned. Once you have isolated, first rule is you have to characterize the isolated bacterial strain. And how do you characterize them? By conducting biochemical characterization or ecophysiological characterization. Once you have characterized it, you can go for direct identification through molecular techniques by 16S rRNA gene sequencing approach. What do you and how do they do 16S rRNA gene sequencing approach? First, what they will do is first they will take your isolated pure culture. They will first extract the DNA. Once the DNA has been extracted, they will look for the 16S rRNA because this is the signature ribosomal RNA, which you can think of like an ID card to the each microorganism. Because if you find a particular signature of the microorganism, then it belongs to the that group. So once you got the 16S RNA sequence, you can use different bioinformatic tools like BLAST and um, phylogenetic tree. You can construct the tree and identify what is the particular microorganism. So this is the 16S rRNA gene sequencing approach. If you want to know more detail, you can visit the week 1 or week 2 lecture where I have elaborated on the 16S rRNA gene sequencing. So this is the broad overview of the culture dependent approach. And in order to set the culture dependent approach, obviously you need the first the resource. What is the resource? It can be either carbon, nitrogen, or phosphorus, or sometimes other nutrients will also, like micronutrients, will also play an important role. And if they need appropriate electron donors and acceptors, you have to provide. And if possible, you just provide a specific substrate to the microorganism to enrich the specific group of organism. And not only the resource, you have to give appropriate condition. It can be a pH, temperature, salinity, pressure, or redox condition. Or sometimes combination of both needs to be played in order to identify a perfect microorganism for your research purpose. Then moving on to the culture independent analysis of the microbial community. First, you have to quantify them using the various techniques like cell staining and microscopic observation. And what are all the other techniques that are available? You can straightly go for general staining method, or you could use fluorescent in situ hybridization. We will be discussing this in the upcoming slides. Then PCR methods. PCR refers to polymerase chain reaction, where you try to amplify the DNA and try to identify the microbial community. Then you can go for microarray and microbial diversity through phylochips, which is more advanced techniques for analyzing the diversity of the microbial ecosystem. And last, you can analyze the environmental genomics and their related methods. So these are all the some of the techniques that are available in the culture independent technique. And as you all know, culture independent technique is nothing but without culturing them. you can identify what are all the microorganisms that are present in a given community. So first we will see the general staining method. What does it mean? That staining involves you will be applying a particular dye to the sample to make the cell structure visible. Once it is visible, you can observe under the microscope. And what and how it is done? First, you will be following a series of steps where you will be applying a primary stain, and then you will be adding a mordant which helps to attach the primary stain into the cells, and you will use a decolorizing agent to remove the excess stain, and finally you will apply a counter stain for the contrast observation. So this is the general method that are using for the staining purpose. And what are all the merits of this staining? It helps in quantifying the microorganism in the natural sample, and they are reliable to some extent, and it can even detect the live cells. But it is not as accurate as the advanced staining techniques, but it gives you some idea on the how this structures are, how the morphology is, whether it is spherical or a rod-shaped bacteria, or it occurs in group or individual. But it doesn't provide any clues on the physiology or phylogeny. What does it tell? It can tell the cell morphology and colony characteristics, but it doesn't tell what is the microorganism. For that, you have to depend on the molecular techniques like 16S or RNA approach for identifying this microorganism. And if you see, there are numerous staining techniques that are available. Now we will see one of the staining technique, which is viability staining. Why it is the word itself tells us it is used to estimate the viable cells. So there is an staining kit is available, which is live or dead bacterial viability kit, which is available in the market. It is used to normally to estimate and differentiate living cells and the dead cells. So it helps in differentiating both dead cells and live cells by staining differently. So this kit helps us to analyze both enumeration and yield viability data. So how does it done? Here we will use two dyes. One is SYTO, which gives the green fluorescence to the cells upon addition, and the other one is propylium iodide, which is red fluorescent dyes. So what is the principle behind it? For example, if you apply first, you have to apply this cell SYTO, which penetrates into the membrane, and it tries to stain all the cells. And next, you will be applying the second type of dye, which is propylium iodide, but it only penetrates the cells which are damaged, because the damaged membrane will have pores into it, so that it goes into the nucleus and stains the nucleus red. And how do we differentiate the dead and live cells? If the live cells are present, you will get a green fluorescence signal. And if the cells are dead, you will get the red color fluorescence. From the right hand side images, you could clearly distinguish between the live and dead cells, where dead cells are red fluorescence, whereas live cells are green fluorescence. And if you could see some orange color pattern, this indicates there is a combination of both live and dead cells. So the cells with compromised membrane will stain red, which indicates that cells are dead, whereas cells with undamaged membrane will stain green. So this is how you differentiate the live and dead cells, which is not seen in the general staining technique, where through general staining technique you could only stain the cells, but you cannot differentiate which is live and which is dead. So this viability staining kit helps us to differentiate both live and dead cells. So if we move on to the next fluorescent dye, which is green fluorescent protein, this helps us to provide a green color to the cells. And what is it? It is a protein that are composed of 238 amino acids. 238 amino acids that exhibits bright green fluorescence when exposed to light in the blue to ultraviolet range. Although many other microorganisms show similar fluorescent proteins, GFP, which is green fluorescent protein, they are first isolated from the jellyfish Aquaria victoria. So what does it mean? It provides green color to the cells which is expressed with GFP protein. So what is this merit? The merit is it is used to genetically engineer the bacteria to make them green fluorescence, and it is also used to track the bacteria present in the environmental sample, and also it acts as a reporter gene. Reporter means it reports the gene whether the gene is present or not. So it acts as a tag to the protein. We will see in the next slide how it is done. So here, if you could see that reporter gene will always be present near to the promoter that you are studying. So once it has been translated to mRNA and reporter protein, the reporter protein will present in the green color, which signifies the presence of the particular promoter to our cells. And from the right hand image, we could observe the green color fluorescence around the cells, which predicts that our gene has been successfully inserted into the microorganism. This can be applied to the cell as well. You could tag the green fluorescent protein to the cells, and if you observe under the microscopy, you could observe the green color microbial cells, which tells us the visualization of the cells under better condition. So this is also one of the fluorescent dye as we seen previously, the propydium iodide, which gives the red color, and SYTO, which provides the green color. So these are all the some of the fluorescent dyes that are available for the staining technique. And next is, if you see the microscopy, there are still the limitation as we have seen that it doesn't provide the phylogenetic analysis of the strain. It only gives the molecular like at the colony morphology level. The first limitation of this microscopy is that prokaryote cells are vary in size. Very small cells can be a major problem and remain unnoticed. What does it imply? Very small microorganism escapes from the detection of microscopy when we are viewing the larger cells. And some cells are near the limit of resolution of the light microscope. So in case of light microscope, blurry limit is there beyond which the details of the cell structures vanishes. So it only operates at the limited limit of resolution. And such cells can be overlooked in the examination of the natural sample, particularly if the sample contain high level of particulate matter or large cells. What does it tell? The tiny cells will get lost when they are mixed with particulate matter or large cells, so that we will lose our information on the microbial cells. And it is often difficult to differentiate the live and dead cells or cells in general form certain inert materials in natural samples. That is, the microscope can show the size or shape, but it cannot tell the genetic relationship between the microorganism. That is, you need a more advanced tool in order to study the phylogenetic relationship between the microorganism. This is what they are telling at the last point, that none of the advanced microscopy techniques tells us the phylogenetic diversity of the microorganism that are present in the habitat. That is, it can tell the morphology of the colony, but not the name of the microorganism, for which we have to use the 16S rRNA gene sequencing approach, where you could identify the organism name. So these are all the some of the general microscopy technique, and we will see the more advanced one, which is fluorescent in situ hybridization technique. What does it mean? It means here also we will be attaching a fluorescent probe to the DNA or RNA, and we make them glow under the microscope. We make them to see the microorganism under the epifluorescent microscope. What happens? It allows for the fast detection and enumeration of specific microorganism. This method, what it uses fluorescently labeled oligonucleotide probes, which is usually 15 to 25 base pair. What happens? This 15 to 25 base pair, they specifically bind to the microbial DNA or RNA that we are studying in the sample. So once it attaches to the microbial DNA, it starts to glow and helps us to identify under the microscope. So it is particularly used for targeting the rRNA. As I said, it is a unique ID code that every microorganism carries. So this is why it is used as a molecular tool for the cultivation independent identification. Not only identification, it also helps us to visualize and quantify the microorganism that are present in the environmental sample. And this approach is based on the fish, not only a visualization, it also applied to analyze the abundance, spatial distribution, and physiology of the microorganism. So what does it tells? It acts as a GPS tracking to know the exact location inside the complex systems. And the general concept is that it is a probe which is 15 to 25 base pair. It can be DNA or RNA probe, but that must be complementary to the DNA or RNA that are studying in an environmental sample. So this is the basic concept of fish where you will be first designing the probe, and this probe will be attaching to the DNA that we are studying. Once it is attaches, it gives the color or glows, and that can be viewed under the epifluorescent microscope. And if you see the advantage of fish over the other microscopy technique, the first advantage is that it reveals the localization of targeted microorganism within the complex environment, such as biofilms. What does it tell? It acts as as I said earlier, it acts as a GPS track. So it tells the exact location even in the complex environment like biofilms. And it helps us to co-localize the patterns of the specific group of microorganism with other microorganism. So it sees and helps us to which microorganisms are standing together. It also helps us to identify and characterize the specific microbe-microbe interaction, whether they are competing each other or have an antagonistic property. So this information can be attained with the help of the fish, and it is one of the most successful technique used to tell where they are, how they interact with, and how and what they are doing in the particular environment. So this is one of the most advanced technique when compared to all other microscopy techniques. And if you see, there are two types of fish that are available. One is ISRT fish, which is in situ reverse transcriptase fish, and other is codfish, which is catalyzed reporter deposition fish. So these are advanced level in the fish. If you see the in situ type, in situ reverse transcriptase fish, it is used to measure the gene expression in a microorganism in a natural sample. So it helps us to measure and look at the gene level that are present in the environment. And next is catalyzed reporter deposition fish. Here, what happens is that the signals that we are getting in the fish are usually a weaker signals. So in order to amplify or enhance the signals, we will be using the catalyzed reporter deposition technique where the signals will be enhanced so that we can see them accurately. Here also our target will be mRNA because this form of RNA is less abundant in the cells when compared to the rRNA. So standard fish techniques cannot be applied. So that in that case, we can apply cord-based fish technique so that the signals can be amplified much greater, because if you want to track the mRNA level, the mRNA among the three types of RNA, tRNA, rRNA, and mRNA, mRNA is less abundant when compared to rRNA. So what happens? The mRNA signal will be very low or weak. So in order to enhance the signal or amplify the signals, we'll be using the cord fish type of technique in order to amplify the signal so that we could observe them more accurately. So this is all for the today's lecture. Now we can move on to the MCQs. Is it fine? We can move on to the MCQs. So this should be an okay. This should be an interactive session. So I will be giving the questions. You just take your own time and post your answers in the chat box, or you can unmute your mic and you can answer the questions also. So till now we have seen the various concepts of what is microbial ecosystem and methods of microbial ecosystem, where we have seen the isolation technique, both pure and enrichment culture based technique. And in order to view that, we have seen the staining technique and different types of staining, and different type of techniques, how to separate the culture and how to quantify them. And also we have seen the limitation of microscopy, and we have seen the advanced levels like the fish fluorescent in situ hybridization technique. So these are all the methods of microbial ecosystem. I hope we can move on to the MCQs now. So you can post your answers in the chat box, or you can unmute your mic and you can answer the questions. So we will move on to the first question. First question is, aerobic respiration yields more energy, and yet many microorganisms cannot perform it. Which of the following is not a possible reason for this? So which is not a possible reason for the above statement, which is aerobic respiration yields more energy, and yet many microorganisms cannot perform it. Option A, the anaerobic microorganism evolved before the great oxidation event and lived in oxygen-free atmosphere. Option B, the anaerobic microorganism live in habitat which are largely devoid of oxygen. Option C, anaerobic microorganisms have specialized to utilize alternate electron acceptor to avoid competition. And option D, anaerobic microorganism requires less energy than produced during the aerobic respiration. And option E, anaerobic microorganisms are more susceptible to reactive oxygen radicals. Among this following statement, which is not a possible reason for the above statement, you can post your answers in the chat box, or you can unmute the mic and you can answer it. And your answers are, your answer, you can also post your answer in chat box so I can view. What could be the answer? Anyone? I could not hear properly. Moonbliss, can you post your answer in the chat box? Option? Option C. Okay. Any other answer? So now we will see the answer. Answer is option D. Anaerobic microorganism requires less energy. than produced during the aerobic respiration. And now we will see why the option D is not possible reason for that. Because as we all know, anaerobic microorganism shall preferably perform aerobic respiration, right? At under what condition? If it is an facultative anaerobes, because there more energy yield is always desirable. And if you see the aerobic mode of respiration produces significantly significantly higher amount of energy than the anaerobic respiration. And this is because the aerobic respiration fully oxidizes the glucose, while anaerobic respiration only partially oxidizes it. That's why anaerobic microorganism requires less energy than produced during the aerobic respiration is the not possible reason. It is not a possible reason for this. So we will see the other options. If you see the first option, the anaerobic microorganism evolved before the great oxidation event and lived in oxygen-free atmosphere. If you see when the oxygen first disappeared in the earth due to the rise of photosynthetic organelle, what happens? The many microorganisms started to adapt to this oxygen-free environment. And whereas in the option B, if you see the anaerobic microorganism live in an habitat which are largely devoid of oxygen. Yes, it is correct, right? Anaerobic microorganism, they know they don't need oxygen for their metabolism. So it is correct, which is devoid of oxygen. And option C, anaerobic microorganisms are specialized to utilize alternative electron acceptor to avoid competition. This is also true, right? Because they are not using oxygen as the electron acceptor. They use alternative electron acceptors. And can anyone tell what are the alternative electron acceptors that anaerobic microorganism
Sulfur,
use? What are the alternative electron acceptors?
sulfur,
Yes, it is correct. Sulfate, nitrates, sometimes H2S and other compounds, oxidizable compounds. So in this way, they generate the energy in the absence of oxygen. And if you see the last option, anaerobic microorganisms are more susceptible to reactive oxygen radical. Yes, is it so? Because oxygen can be highly reactive and they are toxic, particularly to the anaerobic microorganism, which causes oxidative damage caused by the oxygen. So this is why the option D is not a possible reason for this question. I think we can move on to the next question. The next question is, which of these following is a correct order of depletion of electron acceptor in a closed biological system? Which of these is the correct order of depletion of electron acceptors in a closed biological system? Option A. So, which of the following is the correct order of depletion of electron acceptor in the closed environment biological system? Option A, NO3 followed by O2, SO4, CO2, MnOH4, FeOH thrice. So you can view the options and you have to tell which of the following is the correct order of depletion of electron acceptor in a closed biological system. You just take time and observe the options carefully because it seems to be confusing. But if you have studied the electron acceptor, the redox potential concepts that has been covered in lecture 6, you could find the correct option. So you just carefully observe the option and you can post your answers in the chat box if you are not comfortable. And if you are comfortable, you can unmute the mic and tell the correct option. You just take time and post your answers. Among this, which is the correct order of depletion of electron acceptor? Okay, option C. Any other answer? You can post your answers in the chat box also. Option C. Okay. Any other answer? Okay. Then we will see the solution. The option is D. It is not C, it is D. Now we will see the detailed solution. If you have gone through the lecture 6, Professor has discussed various different reactions that are taking place in the biological system, right? And we have discussed some 8 reactions where different electron acceptors has been utilized for the biological system. And based on the del G value, the order will change, right? So if you see first is O2, followed by NO3, followed by Mn, followed by Fe, then SO4, at last CO2 as in final electron acceptor. And how do we arrange them? It is mainly because of the del G value. Del G represent, what is del G? Del G is,
Gibbs energy.
yes, it's Gibbs free energy. So what happens if a del G is negative? What it implies? What kind of reaction it carry out? If the del G is negative,
Exothermic reaction.
yes, it is an exothermic reaction or it is a spontaneous reaction. So spontaneous reaction means you do not want to give an additional energy to this uh, and additional energy to carry out the process, that is reactant converting into product. And if you see here, the aerobic mode of respiration has the highly negative value -125, which tells us it is more spontaneous reaction. You don't need any external energy to carry out this reaction. And followed by the denitrification process, followed by manganese reduction, followed by iron reduction, followed by sulfate reduction, and at last methanogenesis or acetogenesis, where CO2 is accepted as an electron acceptor. So this is why the option D is correct, and you have to be very careful because the options seems to be confusing, but if you could correctly identify what process they are undergoing, you can identify the correct options. You just be careful on selecting the option. So I hope you understand the concept and I will move to the next question. The next question is, under strictly anaerobic methanogenic condition, which could be the preferred terminal electron acceptor? So under strictly anaerobic methanogenic condition, which would be the preferred terminal electron acceptor? Option A, NO3, option B, FeOH thrice, option C, CO2, option D, O2, option E, CH4. So among this terminal electron acceptor, which suits for strictly anaerobic methanogenic condition? And your answers are option C, okay. Any other answer? Option B, okay. Option B and option C, okay. So here the question is, under strictly anaerobic methanogenic condition, which would be the preferred terminal electron acceptor? Now we will see the answer. Here the correct answer is option C, which is CO2. Why it is so? Because CO2 will accept the electron and become CH4 or other reduced organic carbon product. Such reactions are often carried out by the methanogen. So what does it imply? Yes, if it is an anaerobic, option A, B, C, E will option A, B, C will be correct. But here we have given a condition like methanogenic. And from the previous, we could see that under methanogenic condition, CO2 acts as an electron acceptor, right? So that's why under methanogenic as well as the anaerobic condition, CO2 is the preferred terminal electron acceptor. And we will see the other options too. And if you see the other options, which is NO3, FeOH thrice, these are all electron acceptor, terminal electron acceptor for anaerobic respiration. But it doesn't correlate with the methanogenic condition. And CO2 is the correct answer. And O2, as we already know, it is a terminal electron acceptor for aerobic mode of respiration. And CH4, it is a product that is obtained from the methanogenic condition, and it is not the terminal electron acceptor. It is the product. So the correct answer is CO2, which is the terminal electron acceptor under strictly anaerobic and methanogenic condition. So we will move on to the next question. Which one of the following statement is false? Option A, dissimilatory reactions are primarily associated with catabolism. Option B, anabolism involves polymerization of precursor. Option C, ATP is generated during catabolism. Option D, ETC, electron transport chain, is a part of anabolism. Option E, reduction of NO3 to glycine is a part of anabolism. So, which among this statement is false? You can post your answers. And what is their answer? Okay, option D. Okay, ETC is a part of anabolism. Any other answer? Which one of the following statement is false? Option A, dissimilatory reactions are primarily associated with catabolism. Option B, anabolism involves polymerization of precursor. Option C, ATP is generated during catabolism. Option D, electron transport chain is a part of anabolism. Option E, reduction of NO3 to glycine is a part of anabolism. Okay, option D. We will see the answer. Yes, it is correct. Option D, because ETC is a part of catabolism, not the anabolism. For that, you have to understand the two terms. What is catabolism and anabolism? Catabolism means it is breaking down a substance in order to generate the ATP. And we have seen that electron transport chain where ATP is generated through ATP synthase enzyme. Whereas anabolism refers to synthesis of complex molecules from the simpler ones that requires the energy. So here it will use the energy, whereas in catabolism it generates energy. So electron transport chain means it is a series of reaction that occurs within the mitochondrial membrane, and it primarily involved in the catabolism. That is, they generate the ATP by harnessing the energy from the electrons. And why not the other option is false? We will see. Option A, dissimilatory reactions are primarily associated with catabolism. And if you see the dissimilatory reaction, the word dissimilatory refers to breaking down a complex molecule, right? If it is a breaking down of complex molecule, obviously it comes under the anabolism. So dissimilatory reactions are primarily associated with anabolism, not the catabolism. And option B, right, breakdown of complex molecules, so it is a catabolism reaction. Sorry, it is a catabolism reaction, not the anabolism. And anabolism involves polymerization of precursor. What does it mean? So anabolism already know it is a building the complex molecules from the simpler ones. Here amino acids will be polymerized from the precursor in order to form protein. So this is also true. And option C, ATP is generated during the catabolism. It is correct, right? During the cellular respiration, what happens? Here breakdown of energy and a breakdown of organic molecules will be taking place to release the energy such as ATP. And option E, reduction of NO3 to glycine is a part of anabolism. And it is correct because when NO3 is converting into an amino acids, here building of complex molecule is taking place. So this is a part of anabolism. So the option D, which is ETC, is a part of anabolism is the false answer because it is a part of catabolism. So we will move on to the next question. Syntropy is crucial for success of many anaerobic process, primarily because it allows aerobes and anaerobes to cooperate. Option B, it reduces harmful competition between aerobes and anaerobes. Option C, it allows even energetically unfavorable reactions to be carried out. Option D, it allows oxygen transfer between anaerobes and aerobes. And option E, it allows H2 accumulation by the anaerobes. So from this statement, which is suited for syntrophy, is crucial for success of many anaerobic process, primarily because of what is the reason. You can post your answers, and your answers are option C. Okay. Any other answer? You can see the questions and you can take time. Any other answer? Option B. Okay. Now we will see the correct answer. The correct answer is option C, and we will see why. Because the H2 transfer among the centrofic partners, they allows even thermodynamically unfavorable reaction to be broken down into multiple favorable reactions and lead to bioconversion of substrate. So this is the basis of syntrophy, right? What happens in the syntrophy? Different group of microorganisms, they involve in the oxidation of substrate and they convert the unfavorable reaction into favorable reaction by cooperating each other. So it tells us the mutualistic relationship between the microorganism where metabolic byproducts of one microorganism will be utilized as substrate by the another. That is why it allows energetically unfavorable reaction to be carried out by the group of microorganisms. So we will see why other options are false. So syntrophy, what happens? It is normally occurs between the anaerobes, not between the aerobes and anaerobes. So the first option will be eliminated because it involves aerobes and anaerobes. And in second option, it reduces the harmful competition between the aerobes and anaerobes. Here, what happens? It is already a competition between anaerobes, not to the aerobes. So there will be no oxygen presence in the environment. So there will be no aerobic microorganism involved. So there will be no direct competition occurs between the aerobes and anaerobes. And if you see the option D, it allows oxygen transfer among the anaerobes and aerobes. This is also because anaerobes cannot utilize the oxygen, right? Then how the oxygen will be transferred among the aerobes and anaerobes? It will be armed by its present. So there will be no oxygen transfer occurs. And last option, it allows accumulation of H2 by the anaerobes. So here, the H2 accumulation is not the primary benefit. Here what happens, it allows the removal of the accumulated H2 by the another microbes. So here not accumulation, it removes the accumulated H2 by the anaerobes. That is why option C is correct. It allows even energetically unfavorable reaction to be carried out by mutualistic relationship between the different anaerobic microorganisms. So I hope you have understood, and we will be moving on to the next question. So which of the following statement is correctly matched? Option A, who is here, which tells us the system level understanding. Option B, what are they doing, tells us about the phenotypic potential. Option C, what can they do, tells us about the community function. Option D, what are they doing, tells us about the community composition. And option E, what can they do, tells us about the phenotypic potential. So among these statements, which of the following is correctly matched? You can post your answers. You can take time. You can post your answers in the chat box. And your answers are option C. Okay, any other answer? Option C. Okay. Any other answer? So now we will see the answer. The correct answer is option E. Now we will see the explanation for that. So who is here tells us the community composition. Whereas what can they do tells us phenotypic potential, and what are they doing tells us the community function. And if you see the option E, what can they do correctly matches with the phenotypic potential. Whereas other options are not correctly matched. This you can go through the lecture 2 of the environmental biotechnology course. And as we have seen the four fundamental questions, this gives you an idea on who is here tells us the community composition, where it tells us what are all the microorganisms that are present in the ecosystem and what can they do. It tells us what is their characteristics of the microorganism and what are they doing. Tells us about the community function that they perform in a microbial ecosystem. So the correct answer is option E. What can they do? Phenotypic potential. So we will move on to the next question. Which of the following researcher's discovery path is incorrect? Carl Hu's 16S rRNA based phylogenetic analysis, Norman Pace rRNA based phylogenetic analysis, option C, Robert Koch pure culture technique, option D, Sergey culture independent analysis, option E, Staley and Konaka great plate count anomaly. So among these statements, which of the following researcher and their discovery path is incorrectly matched? You can post your answers. Option D. Okay. Any other answer? Option D. Okay. So now we will see the answer. Yes, it is correct. Option D, that is wrongly matched because he discovered chemoautotrophy concept, not the culture independent analysis. Whereas Carl Hughes discovered the 16S rRNA based phylogenetic analysis and Norman Pace rRNA based phylogenetic analysis, where identification of microorganism normally carried out using the pure culture, and Robert Koch discovered the pure culture technique, which indicates the absence of undesired or other contaminants in the culture. And we have seen the great plate count anomaly, where there will be always the discrepancy between the number of bacteria that are involved in the sample using the plate count. So this is discovered by the Staley and Konoka. So among this, option D is the wrongly matched one. So we will move on to the next question. Greater than 99 percentage of all the microorganisms cannot be cultured. Which of the following is not a probable explanation for this? Option A, these microorganisms are not viable. Option B, these microorganisms are dormant. Option C, these microorganisms are mutant of culturable strain. Option D, we do not have laboratory growth or media conditions suitable for them. Option E, these microorganisms cannot survive in the pure culture, that is, without microbial interaction. So, among these statements, which of the following is not a probable explanation for this? I will repeat the question. Greater than 99 percentage of all the microorganisms cannot be cultured. Which of the following is not a probable explanation for this? Your answers are option C. Okay, these microorganisms are mutant of culturable strain. Any other answer? Okay, now we will see the answer. Yes, it is correct. Option C. These microorganisms are mutant of cultural strains. Since unculturable microorganisms form the majority of the microbial diversity, they are expected to be genetically distinct, wild type strains, right? So that is why it is not a probably not the answer. And we know that non-viable microorganisms cannot be cultured. So option A is correct, is probable reason, and the dormant microorganism means they do not grow under the standard condition, so it explains the non-culturable. And option D, if they don't develop a right combination, then we do not have the laboratory, we don't have the correct combination to grow them. And option E, these microorganisms cannot survive in pure culture without microbial interaction. Many microorganism requires other microorganism to grow, right, to make them difficult to culture in the isolation. So this is also a probable reason. So among this, option C is not the probable explanation for the above statement. So we will move on to the next question. Enrichment culture. Enrichment culture is used to option A, enhance the total microbial community. Option B, enhance specific section of microbial community. Option C, selectively allows growth of specific microorganism. Option D, selectively allow bacteria to grow. Option E, visually distinguish the group of microorganism. So among this statement, which is used to describe the enrichment culture. And your answers are option C. Okay. Selectively allow the growth of specific microorganism. So we will see the answer now. Yes, it is option B, enhance specific section of microbial community. So we know that enrichment media itself enrich the specific type of microorganism. So if we see the definition, it is one of the technique that are used in the microbiology to selectively increase the population of a specific group of microorganism within a mixed sample by providing the condition that favors the growth of particular microorganism while inhibiting the others. And if you see the other option, the enrichment culture don't aim to increase the overall number of microbes that are present in the sample, but rather to selectively enrich the specific group of microbial community. So it doesn't enhances the total microbial community, it only enhances the specific section of microbial community. And even though C looks looks similar, but option B is more accurate as it emphasize a section only a particular group of microorganism as enhanced. So C is also not accurate, but B is more accurate. Whereas option D selectively allows bacteria to grow. It is not enrich not just bacteria. It can also enrich other type of microorganism also by providing appropriate condition and resource. And option E, visually distinguish group of microorganism. For distinguishing visually, a differential media is there. So enrichment does not encompass a visual technique, right? So option E is also false. So among this, option B is correct. So now we will move on to the next question. The last question. A medium devoid of organic nitrogen was inoculated with soil and amended with ammonia. After an adequate incubation period, the culture was subcultured to microbial media with and without ammonia. The negligible growth was seen on media without ammonia, though growth appeared on the media containing ammonia. So this could be due to option A, lack of selective pressure for nitrogen fixation, lack of selective pressure for ammonia utilization, catabolite repression of ammonia synthesis, toxicity of ammonia, option E, enrichment of ammonia phase. So, which is the correct reason for this observation? Where a medium devoid of organic nitrogen, you have a medium where organic nitrogen is not present and it was inoculated with the soil sample, but we have added ammonia. So, after some time, the culture was subculture to the media, one with ammonia and the other with without ammonia. But what happens is we see negligible growth was seen on media without ammonia, whereas growth appeared on the media containing ammonia. So what could be the reason for this? Option A, lack of selective pressure for nitrogen fixation. Option B, lack of selective pressure for ammonia utilization. Option C, catabolite repression of ammonia synthesis. Option D, toxicity of ammonia. Option E, enrichment of ammonia phase. You can take your time and you can answer the question. And your answer is option C. Okay. Option C, catabolite repression of ammonia synthesis. Now we will see the answer. Answer is option A, lack of selective pressure for nitrogen fixation. And why it is so? Because adding the ammonia, we observed that growth of microorganisms that are capable of utilizing the ammonia, right? So possibly at what cost? The microorganism has the capable to fix the nitrogen to produce biovaluable ammonia. Hence, removal of ammonia from the media result in population collapse. So that is why the lack of selective pressure for nitrogen fixation is the reason for negligible growth in the media without ammonia. So the correct option is A. And thank you for patiently listening to the lectures and the MCQs. So this is all for the week 7 live interaction session. And if you have any queries, you can ask me through the mail ID ajitvelu284@gmail.com. And if you have missed any of the lecture videos, you can access through my, you can view the videos on YouTube, Ajit Kumar underscore V, or it can be accessed through NPTEL Environmental Biotechnology course as well, where a separate recording session is there where you can access this. videos as well as the PPTs. So this is all for today's. And if you see, we in today we have discussed a very important concepts. First, we have seen microbial ecosystems and their habitat, and we have seen the methods of microbial ecology and what my environmental biotechnology should ask. Then we have seen the important anomaly, which is great plate count anomaly, and we understood the always there is a discrepancy between the number of microorganisms presence and their culturable. And we have seen the categories of culturability, where cultured and uncultured, where in again uncultured, culturable and unculturable organisms we have discussed. And we have discussed the isolation of pure culture of the bacterial strain, where we have discussed direct isolation and enrichment culture technique, and how to select the enrichment culture technique and what are all the conditions that one can use for the process of enrichment culture. Then we have seen the how quantification is done for the microbial biomass. Then we have seen most probable number technique as well, and what are all the other alternatives that are available for the isolation technique. And we have seen the axenic culture, where signifies the purity of the culture that is devoid of other contaminants. And we have seen important qualifying criteria for axenic culture. And we have discussed the culture dependent process and culture independent techniques, where we have seen general staining methods are available, viability staining techniques are available, where we have discussed live dead bacterial viability kit, which is essential for viewing and analyzing both live and dead cells. Then we have discussed other dyes as well, GFP, and how it is helpful for analyzing the gene expression. And we have seen the limitation of microscopy and ended with fluorescent in situ hybridization and two other more advanced fish, which is in situ reverse transcriptase flu- fluorescent in situ hybridization and catalyzed reporter deposition fluorescent in situ hybridization. So these are all for this week, and we will start the bioremediation or biodegradation topic from the next session, and we will see other important concepts and important MCQs that you can that are sample MCQs that you can expect for the end semester as you can attend these lectures as a practice so that you can get a glimpse of the MCQs. So do you have any doubts for this session? And if you have any doubts, you can always mail me in this mail ID, or you can post your queries in the discussion forums so that we can our team and myself will try to solve the queries. And moreover, you constantly watch the videos and try to complete the assignments on time. And I hope you have no queries. Okay, then this is all for the today's class, and you can leave the meeting. Thank you all.